So this is the CAIE, previously CIE, Cambridge IGCSE Biology Paper 2. Now, if you know your paper 2, you know that it consists entirely of multiple choice questions, 40 multiple choice questions. Now, that's easy because the answer is staring you in the face. It's hard because you got to pick the right one, and sometimes that comes down to smart elimination. So let's try to put some of that skill into practice, okay? So, Question 1, a person touches a hot object and rapidly moves their hand away from the hot object. Which characteristic of living organism is this an example of? So the key concept, guys, think, what are they trying to test you here? What on the syllabus are they testing? They're testing the concept of characteristic of living organisms. And based on that, you should know that this is Mrs. Gren. And each of these letters stand for something. And this comes down to revision. So you go to your syllabus checklist, and I've actually compiled the whole thing for you here. So I'm going to show you how you get this in a minute. But you see here on the syllabus, you can have exactly under characteristics of living organisms, MRSGREN. So they're testing on this Mrs. Gren concept. So how do you get this checklist? You go to our website. So open up your browser and you're going to type in www.stoicstudents123.com, and then on the top right hand corner, see those 3 horizontal lines? You're going to click on it, and then you're going to go to study resources. Then you're going to click on syllabus checklists. Then you're going to click on whatever subject you're working on. So we're doing biology today. And then you find your syllabus, so this is CIE, we click on that, and then you can download the whole thing. This is exactly the same as the exam board. I've just compiled it in a really organized way, and for each of the syllabus, I've put down on the right-hand side sort of like a checklist, a traffic light system, so you know which concept you're confident and which one you need to spend more time on. So you're going to go through this whole thing as you revise, make sure you don't miss anything, and therefore you're fully prepared for the exam. So keep track of your own revision. Okay, so coming back to this question now, it says, which characteristics is this an example of? So if they're moving their hand rapidly away from the hot object, that's definitely not excretion, okay? Because excretion is what? Excretion is when you are urinating, right? So you're getting rid of waste. The waste is CO2 and urea in your body, and this is not excretion. This is definitely not growth because he's not growing anything. It's not reproduction because no one is doing sexual or asexual reproduction by moving their hand away. So it's basically sensitivity. It's sensitivity. So that's the answer. Okay? Next, which types of organisms are used in genetic modification because they have plasmids? Okay, now, really, guys, the word plasmid is only associated with one thing, and that's bacteria. Only bacteria has plasmids. Nobody else does. Just remember it. Question 3, the list shows some of the structures contained in cells: cytoplasm, cell membrane, cell wall, and chloroplast. And how many of these structures are found in the cells of all living organisms? All living organisms. So just have a think, right? What is the most basic thing you need for a cell? Like the most basic thing, if I told you to build a cell, what would you have? You're going to be like, okay, I'm going to start with this outline, which keeps everything together, and this is called the cell membrane, right? And then you're going to be, there's going to be stuff inside the cell, right? And this stuff, this sort of fluid that contains everything, what is that? That's the cytoplasm. So you definitely need to have these two. Do you need to have a nucleus? No, you don't, because bacteria are prokaryotes and they don't have a nucleus, right? Do I need to have a cell wall? No, only plant cells or bacterial cells have a cell wall, right? Do I need chloroplast? No, I don't need that. That's only for plant cells. So therefore, the only 2 things that all things must have is the cell membrane, because think about it, guys, it's like the outline. So how can you build a cell without an outline, right? It's like you can't build a house without walls. And then you have the cytoplasm, which is the space that's inside. So therefore, it's just 2 things that all cells have, just these 2. Okay? Okay, and then question 4 here says a student draws a red blood cell. The diameter of the red blood... Okay, before I proceed further, I'm just going to draw everything out because I like to visualize it. So here's our beautiful red blood cell, and I'm going to give it the biconcave shape that it is famous for. And then Now, what does it say? The diameter of the red blood cell is 20 millimeters. So this is the diameters, right? This is 20 millimeters. So I'm visualizing the question as I read it. And it says the actual diameter of the red cell is 0.008. So obviously the actual one is much smaller, right? You can't see it. That's why you need a microscope to see it. And it's asking you, what is the magnification of the student's drawing? So he would have magnified this red blood cell. So you need this magical formula called MIA. Okay, M-I-A or A-I-M. So here we go. You can choose how you want to memorize it. Let's do A-I-M. Okay, AIM, because our aim is to get full marks. Okay, A-I-M. So they all stand for different things. A stands for the word actual. So the actual size. Okay, let's scroll up a little. The actual. M stands for the magnification. M is the magnification. And then what is I? What would be your guess of what I is? I is the image, is the thing that he's drawing here, is the image. You've got to make sure you know what everything is. Once you know what everything is, you do your magical maths, which is you rearrange the equation so that you have an equation with the correct subject, right? So what is this correct subject here? Tell me, what is the thing we're trying to find? What are we trying to calculate here? Can somebody tell me? What are we trying to find? They're asking for the magnification, right? So obviously it's going to be M is equals to I divided by A. Does everybody agree on the same page, right? M equals to I. That's what the triangle means, guys. You understand, right? The M is equals to I divided by A. So similarly, A is equals to I divided by M, right? And I is equals to A times by M. That's why it's a triangle. So M equals I over A, and then you're just going to do the calculation. Before you do the calculation, please check that the units are the same and everybody is in what? Everybody is in millimeters, and therefore it's okay. I can proceed. So let's plug in the number. So this would be 20 divided by, what is the A? A is actual, which is 0.008. So please whip out your calculators. 20 divided by 0.008, that would give me 2,500. Please let that be one of the options. Yes, it is. So it's D. Okay, now I have more practice on magnification questions. If you go to my channel, you can look for this video, and it's called magnification practice. Okay, so try that, and there's more exam papers on that topic in there. Okay, question 5. Samples of plant cells from the same species are immersed in 3 different liquids. And that would be this one, and this one, okay? And so it says the low concentration sucrose solution has a high water potential than the plant cell cytoplasm, and the high concentration sucrose solution has a lower water potential than the plant cell cytoplasm. So guys, if you ever read a question that's too long for your brain, start again and then draw it out as you're reading it, okay? So let's do this. Now, the first sentence here, it says there are 3 different liquids, okay? So 3 different liquids. I'm going to draw that right now. 1, 2, 3. So visualize it. I'm a visual learner, so let's do that. Now, here it says the low concentration sucrose solution, so this one here, has a higher water potential than the plant cell cytoplasm, right? So there's a plant cell cytoplasm. Well, there's a plant cell in each of them, I assume. Okay, because here's the plant cell, right? Here it is, the plant cell, here, here, and here, right? And this blue one in the middle has a high water potential. And then you want to start thinking about what that means, right? Because water always moves from high to low water potential. So since it's high on the outside, how's the water going to move? Tell me, is it going to go from the outside to inside or vice versa? Again, it goes from high to low, right? So it's going to go from the high outside to the low inside. So this is the osmosis direction. Okay, and then let's do the next one then. So for this, it says the high concentration sucrose solution has a hello, etc. So this is the high concentration sucrose solution as this one here, and it has a low water potential outside, which means that it's not very high, it's low. Water, therefore, has a higher potential on the inside. So high inside, low outside. Water moves from high to low water potential always. Therefore, it will move out of the cell. Agree? From high to low. From high to low. So therefore, the water is going to move out like that. Now I want you to think about what the cells actually look like now if water has moved in versus moved out, right? So in the blue test tube, if water has moved in, that's like that cell is now going to get completely filled up with water. So it's going to be like really fat and pudgy. Really fat and pudgy. And that pudginess we call turgid. We call turgid. And therefore, I'm going to write this down here, okay? So it's really, really clear. Turgid. And that now we start to eliminate. That means the blue one can only be turgid. So it's got to be this one or this one. Therefore, it's either B or D. So we've already eliminated A and C. Let's get rid of that right now so it does not distract any of us. So forget about A and C, okay? Then we think about the other one. Think about what happens in this pink one. Now, since water has left it, look, if water has been sucked out of it, what is that cell going to look like? It's going to be really sort of like, flat, and because water has been sucked out of it, right? So in that case, this is called plasmolized. Okay, plasmolized. And therefore, the answer would have to be which one? Well, they're both plasmolized. Oh dear, so we still are nowhere clear, okay? So then we have to think about this one then. So for this one, what would be the state of the cell? Would it be turgid or flaccid? Turgid or flaccid? Well, you got to think. This one is distilled water. That means it's nothing but water. Tons of water here. So how's the water going to flow? How is the water going to flow? It's going to flow from out to in as well because there's so much water. It's just distilled water there, isn't it? And since that's the case, then your cell is also going to look turgid, isn't it? It's still going to be very, very, very turgid. So it's going to be pudgy, like a baby's cheeks. And therefore, the answer is D. Okay? Right, question 6, it says, which process requires energy from respiration? Please, I hope this is straightforward to you now. Active transport, right? Because let's do a bit of revision here. These two are completely passive. Passive means no energy required. This one is active because, surprise, it's called active transport. So what are the key words? It requires energy in the form of what? What is it called? It's called ATP, isn't it? And it's definitely got nothing to do with transpiration. That is the movement of water and the evaporation of water along the plant and so on. So that's got nothing to do with respiration. So elimination takes you down to just A. Okay, question 7. The diagram shows the sticky end of the DNA of a plasmid that has been cut by a restriction enzyme. The unpaired bases are labeled. Okay, the unpaired label. So what is the correct sequence of the missing bases? Okay, so do we know how we pair things up? Do we know? A always pairs with T. Someone told me to remember that as apple tree. So A and T, apple tree. Okay, so you can remember that. And then what does C always pair up with? With G. So once you know that, then you can start to do the magic, right? So C, so let's pair each of these up. Let's do it, okay? So this is called complementary base pairing. It means they always pair with the complement. So the complement of C is G. A is T. G is C. T is A. And G is C. So I'm looking for somebody that is G, T, C, A. And that would be the answer C. Easy. Okay, last question on this page. It says a test was performed on a food substance. A positive result was shown by a color change to blue-black. What solution was used for the test? Okay, elimination, guys. So you got to know all of it, basically. So what is it? Let's revise everything. What they test for, and then the color change. Right, so let's do it. For burette test, it tests for what? Protein. And it goes from what? It goes from blue to what? It goes from blue to purple, although everything's looking pink right now. Now, DCPIP, what does this test for? It tests for vitamin C. Vitamin C, right? And what is the color change? It goes from blue to colorless. Blue to colorless. If vitamin C is present, right? Iodine solution test for what? Thank you. Starch. And what is the color change? It goes from brown, because iodine is brown, into blue-black. Blue-black. Right? And then finally, Benedict solution test for sugar, such as glucose. And it goes from blue to what? Blue to brick. Red, precipitate. And therefore, who is turning blue-black for us today? It would be this one, and therefore I'm testing for starch, and that is definitely going to be iodine solution. So the answer is C. So question 9, it says a student investigates the breakdown of fats in milk by lipase. 4 test tubes labeled A, B, C, and D are set up. The table shows the contents of each test tube. And in which test tube will the contents become acidic most quickly? Okay, so let's talk about the breakdown of fats. Again, let's hone in on the key thing that they're testing on here. So when you're breaking down fats, what are 2 things that are required? I need 2 things. I need 2 of them. I need, number 1, I need an enzyme, which is lipase, right? Lipase breaks down lipids, which is basically fats. So that's my enzyme. But that is not enough, guys. I also need bile, because the bile will help to emulsify the whole thing, so therefore making the digestion of the lipids easier. So I need lipase and I need bile. So I need both things. So therefore, in my test tube, for anything to happen, I definitely need the bile. Right? So I need anything, so that therefore this eliminates C, because C doesn't even have bile. Forget about it. You cannot successfully digest fats and lipids without bile. I also need lipase, right? So I definitely need lipase. So that eliminates which one? That eliminates B, because B doesn't have lipase. So now I've narrowed it down to A and D. Now, the question says it's investigating the breakdown of fats in milk. So, of course, I need milk, right? So, D doesn't even have milk. D doesn't have milk, so I'm going to eliminate that. And therefore, this is A. And if you're wondering why there's a test tube here, it's because I'm trying to show what's happening, okay? So, you have the milk in there. You have the milk in there. And then you're going to add the lipase and the bile, right? They both go into there, and therefore, they will start to break down the things, and therefore become acidic. We didn't talk about that, guys. This is good for revision. Why would it become acidic? Why? Because when you break down lipids, what do they become? What are the building blocks of lipids? Lipids are made up of, remember, like this. Does this mean anything to anyone? Right? What does this mean? It means it's made up of this pink unit here, which is what? What is it? It's the glycerol. Glycerol. And what are those 3 legs there? What are they? Those 3 leggies. Those are the fatty acid tails, right? The fatty acid tails. These are the fatty acid tails. Fatty acids. So 3 fatty acid tails, right? And that's why it becomes acidic, because you've now broken down so that you release those fatty acids. That's why it's acidic. So before you break it down is this whole thing here. That's not acidic. But after you break, right? You break it with the lipase. It will then become acidic because the fatty acid tails are now released. Okay, question 10 now. The diagram shows an experiment to investigate the effect of increasing light intensity on the rate of photosynthesis in an aquatic plant. The rate is found by measuring the rate of bubble released by the plant. Okay, and think about what the bubbles are, right? Why would it be releasing bubbles? What are those bubbles? What gas are those bubbles? Because it's doing photosynthesis. What is the gas that's released in photosynthesis? Oxygen, right? So this is nothing to do with the question, but really it's just to make sure you understand the question, right? Make sense of it. Oxygen bubbles. So as the light intensity increases, the rate of bubble release increases and then becomes constant. And the question says, which statement describes why the rate of bubble release becomes constant at high light intensities? Okay, so let's try to make sense of things, try to eliminate again. Right, so B doesn't make sense because light intensity would never be a limiting factor because it continues to increase, right? It goes all the way to high light intensity. So it's not a limiting factor. Limiting means it does not increase anymore, so it's not B. C, oxygen availability becomes a limiting factor. Not really. This is photosynthesis. Photosynthesis does not require oxygen. It produces oxygen. So therefore, I don't need oxygen to be available. And then water availability becomes a limiting factor. It doesn't look like water is going to be limiting anytime soon because the whole thing is dunked in water, right? So it's not going to be that. And therefore, the answer is A. And just make sure you understand carbon dioxide concentration becomes a limiting factor, right? Because if you don't have enough CO2, which is one of the reactants, remember, guys, photosynthesis, right? CO2 plus what? Plus water makes oxygen and glucose, right? This is the equation for photosynthesis. So there's plenty of water. So the other thing that is limiting is CO2 concentration. CO2 concentration, that is a limiting factor. That's a key word, limiting factor. And when it's limiting, the rate will become constant because it's not going to be increasing anymore. Okay, question 11. The diagram shows a section through a leaf of a flowering plant. And it asks, where does photosynthesis take place? Okay. So let's just label everything for here. I love labeling because it just helps me to revise everything. So there's like a couple layers here, okay? There's this layer, there's this layer, there's this layer, and there's this layer. There's 4 layers really, okay? So this is called, the green layers are called the epidermis. So there's upper and lower epidermis, okay? And then the purple layer is called the palisade, mesophyll. And the pink layer is spongy because there's lots of air pockets inside, okay? Spongy mesophyll. So let's just know the layers first. That's very helpful. There's another thing I'm looking at that's very, very good to revise, which is like this big chunk here. Do you see how the cells here are really concentrated compared to everywhere else? You see that? So what that is called is a vascular bundle, okay? Vascular bundle. What does it contain? 2 things. It's a bundle of what? Bundle of xylem and phloem, these 2 things, which carries waters, minerals, and energy for the plant, right? Sugars and so on. So this is sort of everything that's here. Now the next thing you want to focus on is the actual question, which is where does photosynthesis take place? Now photosynthesis takes place in the places where there is chloroplast or chlorophyll. That's very obvious, right? Because photosynthesis requires chlorophyll. So where are all the chlorophyll? Where is it? Now, if you look at these are plant cells you're looking at. You see all the things I'm outlining, these are all the plant cells. And what do all the plant cells have? Do you see that? It always has a little circle, just one little circle, this white circle. So that one little circle is the nucleus. And then it's got a big thing. What is that? That is the vacuole, right? Because plant cells have vacuoles a lot of the time. So you see everybody's got one nucleus and one big vacuole. What else do these plant cells have? These black things. These black things, or little dots, those are the chloroplast. And it makes complete sense that the palisade mesophyll layer of cells have a lot of chloroplasts. Look how many black dots there are here compared to the cells down here, which only has 1 or 2, right? And that's because the sunlight is beating down from the top. So of course you want to have the palisade mesophyll, the first layer, the top layer, to have lots of chloroplasts to catch that sunlight in order for photosynthesis to happen at a maximum rate. So it all makes sense. But anyway, who has the chloroplast then? 1 definitely has the chloroplast. So that's got to be either A or B, right? Not C, not D, forget it. 2 does not have chloroplast. There's no black dots in 2. And definitely not 3. And 4, yes, absolutely. So 1 and 4. Therefore, the answer is B. B for boy. Okay, last question on this page. The diagram shows part of the alimentary canal and associated organs. Which labeled part stores bile? Whoa, okay. Again, my favorite thing to do is labeling. Let's pick a lovely color. Let's do pink right now. So A is the stomach. B is what? Ooh, the one that looks like a leaf, guys, it's always the pancreas. Please memorize. The pancreas looks like a leaf, okay? C is the gallbladder. And then D is the liver. The liver is always like this triangle shape. So if you see this triangle, that's the liver. Okay? So that is everything here. And then you want to think about the function, because the function is what you need in order to answer the question, because it will tell you who stores the bile, right? So stomach, just give me 1 word. If someone comes, like an alien comes to Earth and asks you, well, what is the function of the stomach? You give them 1 word. That 1 word is digestion. Okay, so function in green. Okay, function. Digestion. Pancreas is also for digestion. It also produces insulin, which has nothing to do with digestion, but that's not the point of this video, okay? So pancreas, for now, for the digestive part, it produces digestive enzymes, therefore it contributes to digestion. Now the liver doesn't do digestion, okay? Because it's nowhere near the food. It's completely separated. The alimentary canal is like this whole tube that goes from your esophagus down to your stomach, down to your, your small intestine, large intestine, down to the rectum, and then the anus and out, right? That's the whole canal. That's why it's called a canal, that goes on and on. So the liver is completely separated from it. Do you see that? So it doesn't actually do any direct digestion, but it produces something very important that contributes to digestion, and it produces what? Produces bile. Okay, and once that bile is produced, which is here, it's going to travel down here and get stored in here. So therefore, the gallbladder stores the bile, and that's why I'm talking about this in so much detail. It's just to basically circle C as the answer. Question 13, the list contains statements about villi, and you're supposed to find which two statements are correct. So let's just look at what the villi actually is, okay? So you have the human body here, and then you have the digestive system, specifically the small intestines, and then this is the large intestines. Always looks like a question mark, right? So the small intestines, if you go and zoom in on them, so over here, you will have this inner surface there, which is not smooth, but looks like that. And one of them is here, and that is a villi. And sometimes they even talk about the microvilli, is that on each villi, it is also covered in more villi, so therefore they're micro means small, so they're like small mini villi, so microvilli. Now after we look at this, now you want to think about why it looks like that. There's no reason it will look like that unless there's a purpose, and the purpose is you have to know what the small intestine does, right? So what is the function of the small intestines? One word: absorption, right? The small intestines does absorption, and if it does absorption, let's zoom out a little bit. If it does absorption, that means you want a large surface area to do that absorbing, because larger surface area makes the absorption more efficient. And based on that, we can already tick number 1, because the villi increases the efficiency of absorption of nutrients, which is whatever you're eating. Number 2, this is definitely wrong. It's wrong because the villi is not in the alveoli. As simple as that, right? The alveoli is in the lungs, and we're talking about the small intestines here. Number 3, it says villi provide a large surface area. Absolutely, that's why they look like that. And then finally, villi filter urea from the blood. It's completely wrong, because it's got nothing to do with urea. Urea filtering does not take place in the small intestines, okay? Urea is a waste product, so that's an entirely different organ system. And therefore, we'll be going for 1 and 3, which is B. Okay, question 14. The diagrams show carbohydrate molecules as they pass through the alimentary canal. Your alimentary canal is basically your digestive system. So from your esophagus to your stomach to your small intestine, large intestine, rectum, and then out through the anus, right? And it asks which row represents the molecules correctly. So just think of the big picture here. You might get thrown off by all the diagrams, but the idea is that as you eat food, those big chunks of food get broken down and broken down and broken down further until they're small enough to be absorbed into the bloodstream, where they will go to all the cells to do respiration, right? Because it's glucose. Now, which diagram, ABCD, looks like things are getting broken down and broken down and broken down? Which one? B. Okay, done. Nice and easy. Question 15, it says, which row shows correct information about 3 digestive enzymes in humans? Now, these are the 3 enzymes here: lipase, maltase, and trypsin. Enzymes usually end with A's. Okay, so you know they're enzymes. And I just want to go a step further, because when I read a question, I just want to make sure I know everything about it, right? So this lipase enzyme here, what is it breaking down? And what does it break down the thing into? So it's breaking down lipids. You can pretty much guess that, right? Lipase, lipids, right? It breaks down the lipids into what? So the lipid is broken down into what are lipids made of? They're made of glycerol. I'm gonna scroll down here a little bit. Glycerol and fatty acids, right? So fatty acids. That's just a bit of revision there in general. And then let's talk about maltase then. Can you tell me what maltase breaks down? Maltase breaks down maltose. Okay, and maltose, if you remember, are 2 molecules like that, and it breaks it down into what? Into glucose. So individual molecules of glucose. So that's what's going on. Okay. So anyway, let's sort of answer the question now. It says, which row shows correct information about the 3 digestive enzymes in the humans? Now, I have this up here for a reason. It's because on my channel in the anatomy series, I did a whole video on digestive system. I go through everything in detail there, where the enzymes, the digestive enzymes are produced, what they break down. It's a very comprehensive video. So please look at that if you want more information. But for this one here, lipase. So you have to know where it does the action, okay? So where it actually acts. So lipase acts in the small intestines, okay? It acts in the small intestines. Maltase, what maltase does is it also acts in the small intestine. This is just one of those things that you have to sort of memorize. And then trypsin acts in alkaline conditions. It acts in alkaline conditions because it's I explain it in this video, so I encourage you to watch that because there's a lot of stuff to revise for this one question, okay? So this is just one of those things you have to memorize, where the enzymes are acting and what conditions they work well under, acidic or alkaline conditions. So that's number 15. And number 16 says, which substance found in a healthy diet helps to prevent constipation? I hope everyone knows what constipation is. It's when you have difficulty for lack of a better word, pooping, okay? So how can you make yourself poo better, basically? And I don't know if a doctor has told you this, but usually if you're constipated, they tell you to increase having 2 things. Number 1 is fluids, drink more water, because if your poop is dry, it's hard to push it out. And then the second thing is have more fiber. And fiber is usually from vegetables, like leafy vegetables, okay, or just vegetables in general. Fruits, vegetables, they give you a lot of fiber. That's why it's important to have that in your diet. You can kind of eliminate the others here because vitamins and minerals are way too small to have any impact on how your poo is formed. So how the fiber works is that it just bulks up the poop, and therefore it's easier to push it out of the rectum and the anus, right? Whereas minerals and vitamins, they're really tiny little molecules, and they don't bulk up the poop in any way. Fat doesn't do anything like that either, so it's fiber, so have more veg, basically. Okay, 17. When is the transcription rate of a well-watered plant highest? Okay, this is a great question. I want you guys to visualize this. First of all, for those that haven't revised, transcription is basically if you have a plant like this with a leaf, let's say, and transcription is when, the official definition is the evaporation of water out of the plant. So basically over there through the stomata, right? But how would the water even be in the leaf? It's because it's been going in through the roots, right? This is water movement. So you imagine the whole journey goes through the roots into the xylem, roots and then root hair cell and then into the xylem all the way up and then into the leaf and then out through the stomata. So that's transpiration. It's basically the evaporation of water from the leaf. Now, when is it the fastest? Now, I want you guys to think of it like this because it becomes really easy. Imagine you have like a t-shirt, okay, that you're trying to dry and you've hung it up outside. So what kind of condition is going to make this t-shirt dry the quickest? Because the water leaving the t-shirt is similar to the water leaving the leaf. Get it? So you want it to be a hot day because that will dry things out faster. You want it to be nice. You want it to be dry or wet if you want your t-shirt to dry. You want it to be a dry day, right? Because if it's really humid, your t-shirt's not going to dry. And then finally, you definitely want a windy day because it will just blow the water molecules away. So just by thinking it like this, you can answer the question, which is C. Okay, great. And finally, 18, it says, which statement is correct for a flowering plant during photosynthesis? So photosynthesis, you have to think about the equation, which is what? H2O plus CO2 makes oxygen, and C6H12O6, which is glucose, right? And then the glucose will then be turned into sucrose, which is then transported around the plant in order to provide an energy source. That's why plants don't have to eat, and they don't have to go shopping for food, and they don't need to cook, because they can make their own energy source, which is the sucrose, which comes from the glucose in photosynthesis. And the answer here would be C, because the leaves are a source of sucrose. Why? Why is that? Source means where it comes from, right? Why is the sucrose originally coming from the leaves? Think about it. Why not in the roots? Why in the leaf? Which part of the plant will be doing most of the photosynthesis? Photosynthesis requires light, and the leaf is very adapted to capturing light. That's why it's big and flat and got lots of chloroplast, right? And therefore, the leaf is going to be the main source of where sucrose is produced. Yeah? And then after that, you want to transport it around the plant, and the sink is where it's deposited. It's usually in the fruits. Okay, so that is the answer for this one. Okay, so question 19. What is an advantage of having a double circulatory system? Now, do we know what this means? This is the circulatory system humans have, and a lot of mammals have, is a double circulatory system. So what it means, circulatory means, how the blood circulates around the body, right? Oxygenated blood, and then becomes deoxygenated blood, and so on. That's the circulatory. Double means, a lot of my students don't know what double actually means. Do you know what double actually means? It means in one cycle of blood going around the body, it goes through the heart twice. Double. Twice. And I can show you that right now. I want you to follow what I'm going to draw, okay? So let's start in the heart. Now, if you still remember the heart, which side of the heart has the oxygenated blood? Which side of the heart? Remember, this is the right side because it's the person's right hand, right? And this is the left side, okay? So which side of the heart has the oxygenated blood? It's going to be the left side. So this side is the red color, which is the oxygenated blood, okay? Oxygenated blood. Now let's pick an organ for this blood to be pumped to. Let's say we're going to the kidney, okay? So here are the kidneys, okay? So let's now start the game. Now, this blood is going to leave the heart. Let's get this bigger, shall we? So it's going to leave the heart and through the aorta, like there, and then it's going to get pumped up, and then actually goes down like that into the kidney through the renal artery. Remember, artery pumps blood away from the heart, right? And it goes into the kidney, which is renal. So it goes into the kidney via the renal artery. And then when it goes to the kidney, why are we pumping oxygenated blood again, guys? Why? Keep asking yourself why, because it'll make you understand and make you enjoy science more. Why are we pumping oxygenated blood? Is because the kidney cells here, they need oxygen for the magical equation of respiration. They need a respiration, which requires oxygen and glucose, right? So the oxygen is coming from this oxygenated blood right now, goes to the kidney, drops off that oxygen, so the kidney can do respiration. And then respiration produces what? Produces carbon dioxide and water, right? Carbon dioxide is the waste. So that carbon dioxide now is going to get dumped back into the blood because I have to get rid of it, right? So that CO2 now goes back into the blood, leaves the kidney through the renal vein. Remember, vein goes to the heart. Veins carry blood to the heart. So it's going to leave the kidneys, and then it's going to go back up to the heart, to the right side of the heart now, because the right side of the heart carries deoxygenated blood. So back up through here into the vena cava, and then into the right heart, and then it's going to pump that. Think about, how do you get rid of CO2? Through the lungs, right? So it's going to pump that deoxygenated blood to the lungs via the pulmonary artery, and then you can breathe out that CO2. Understand? Now, in this thing I've just drawn, how many times did blood go through the heart? It went through once, the red blood, and then it went through again, the blue blood. Do you see that? So this is just one cycle, but it's gone through the heart twice. That's why it's called double circulatory system. So please remember that, okay? So once you remember that, now you try to appreciate why does it do that. Because the deoxygenated blood and the oxygenated blood are separated. See, see, this side is oxygenated, this side is deoxygenated, right? They're kept separate because it's the right side and left side of the heart. And by keeping it separate, you can have a very efficient way of delivering oxygen to the tissues. So that is the reason why you have a double circulatory system. So that's the answer. Okay, so the blue color here is deoxygenated blood. Deoxygenated blood. Okay. Question 20 here. Question 20, it says, the diagram shows the external structure of the human heart. Which part is blocked in coronary artery disease? This is one of the top killers. The top killers in the world in terms of diseases. So it's very good to know about this. And when you hear people say, oh, someone had a heart attack, it is usually due to blockage in the coronary artery. So let's label all of this. Labeling heart is one of my favorite things to do. So A, what is A? It really helps because the heart is pumping blood, guys. It really helps to appreciate like the direction that the stuff is moving in, okay? The blood is moving in. So A here, the blood is moving into the heart, and specifically into this compartment, which is the left atrium, and it's going into there like that. And since the blood is going to the heart, it's going to be a vein. And this is the pulmonary vein. So that's the first thing that's labeled. And then we can label D. D here, that is pumping blood out of the heart. Let me use a different color just to kind of delineate everything better. So this is pumping blood out of the heart, and if you look at this very carefully, it goes this way, but then it splits into two branches, right? And that's because it's going up to the lungs, and you got two lungs, right? Left and right lung. So since it's going up to the lungs, you know, it's going to have the word pulmonary in there. And since it's carrying blood away from the heart because it's going away, that will be an artery, right? A for artery, A for away, so pulmonary arteries. That's what D is. And then we need to know what C is. So what C is that it's actually carrying blood up this way into the right atrium, actually, the right atrium, and this is a vena cava. It carries the deoxygenated blood. So actually, it's exactly this vessel here. It takes the deoxygenated blood back to the heart into the right atrium. Okay, and finally, that leaves us B, which are these very thin. I should really put this in green so that you guys make that association. So coronary heart disease is B here. So if you see this very thin blood vessel right there, that is the coronary artery. And when those are blocked, I encourage you to think about what happens. So it's like this very thin there, right there. So if this is blocked, let's say there's a blockage here, right? Then the oxygenated blood, which is supposed to go down this way to supply this bit of muscle of the heart, so that the heart can get replenished with oxygen and therefore be able to do all the pumping for your entire life. Yeah. Now, if this is blocked, then the oxygenated blood cannot go down, and therefore downstream of this, all the muscles that are supplied by this part of the coronary artery will starve from oxygen. They'll be starved of oxygen, and therefore the heart tissue will die. And if it dies irreversibly, that is a heart attack. And this is why this is called a coronary heart disease. It's when that blood vessel becomes blocked. And it's good to note, guys, why it's so common. Why do you think it's so common, huh? Why is coronary heart disease so common? Look at how narrow this coronary artery is. Look at how thick the other arteries are, right? Look at this pulmonary artery, for example. Look how thick that is. That's not really going to get blocked ever. But these ones are so thin they can get blocked, and therefore that is a coronary heart disease. Okay, and in case you're thinking how to prevent it, or what is blocking it, usually lipids or cholesterol, which comes from your diet. So please have a healthy diet and do lots of exercise, and the arteries in your body will be healthier. Okay, and then finally we have 21, which is one of the symptoms of the disease cholera is diarrhea. That's watery poop that you're getting out of your body. This is due to water loss by osmosis caused by the cholera toxin. Now, what is the effect of cholera toxin that results in diarrhea? Now, this is one of those questions where I'll tell you the answer first and I'll explain it, okay? So this is C. So what happens is you have your small intestine. That's your small intestine right here. And remember all the villi, right? This is the villi that we actually looked at earlier on. So here's all the villi. Okay, so I'll label that as the villi. Now, have a really idea what's going on. This is inside the gut. So this is where all the food is traveling through. And then what you have in the villi is the blood vessels. Why? Because you're trying to absorb the food, the nutrients into the blood vessels, right? So I'm going to have here's all the blood vessels. In fact, let's be more accurate. It's very, very thin blood vessels. They're called capillaries, right? So just have an idea what's going on. Usually in a healthy person, you want to have the food that you just eaten, which gets digested and broken down, and it'll go from the small intestine into your blood vessels, and therefore get carried to everywhere in your body for respiration, because it's the glucose. A lot of the glucose is required. Yeah. Now, what's happening in diarrhea is there's a secretion of chloride ions into the small intestines. So let's go even deeper. And let's have chloride ions as green. So what happens is the chloride ions are originally like here, but they end up here. Okay, so they get secreted into the small intestines. So now have a think about this compartment, which I name A, and this compartment, which I name B. Which side has a lower water potential? Remember, lower water potential means there's less water. Okay, just less water. It's got to be the compartment which is more concentrated by the little green dots. So which one is more concentrated by the little green dots? A. So therefore A has a lower water potential. How does that cause diarrhea? Because water always moves from high to low water potential. So this is high water potential, this is low water potential. So what happens is your water would end up moving from the bloodstream from B to A, and therefore there will be lots of water in your gut. And there's lots of water in your gut. What does your gut do? It produces poop eventually, right? And therefore you poop out that watery poop, which is diarrhea. Understand? So that's why C is correct, because it says you're secreting the chloride ions into the small intestine, which lowers the water potential. And when the water potential here is so low, so this compartment here, guys, the low water potential compartment, here, lower water potential is here, right? Therefore, the water is going to move into the yellow compartment, and then you end up with watery diarrhea. So that's how you can explain how cholera works to somebody. Okay, I like this page because all the questions are fairly straightforward. So it says, which statement about immunity is correct? Let's do a bunch of elimination. So it says, a part of the antibody molecule has the same shape as the antigen as the axon. This is wrong because it would not have the same shape, guys. It would have what we call a complementary shape. It means that it should be a complementary shape. It means that if the antibody looks like this, then your antigen should look like this. So the complementary bit is like this bit, right? So imagine if they have the same shape, guys, then what? You're gonna have an antibody that looks like that, and you're gonna have an antigen that looks like that. How are they gonna fit? They're not gonna fit, right? The whole idea is that they fit, and therefore the antibody can act on the antigen. So the relationship between the shape of the antibody and the antigen is complementary. That is a key word. It's definitely not the same shape, and therefore it is not A. It is not A. Let's look at B. Babies develop passive immunity from the antibodies they receive in breast milk. Absolutely. Do you understand all the words here, guys? Passive immunity means the baby didn't do anything. That's what passive means, right? You don't need to do anything. The opposite of passive is active. So it's passive because the baby does not have its own immunity yet when it's first born. And that's why breastfeeding is so important, because the mother has a lot of antibodies already and passes on those antibodies to the baby through the breast milk that the baby is drinking. So this is a correct statement. This is a correct statement. Okay, so this is correct. But let's keep on eliminating because we learn a lot from just ruling things out. So phagocytes produce antibodies. What is wrong with this statement? Can someone tell me? Do phagocytes produce antibodies? No, they do not, right? It really, who produces antibodies? It should be lymphocytes. What do phagocytes do? They do phagocytosis. They do the engulfing of the antigen, right? So therefore, it is not C. And then D, vaccines contain weakened antibodies? No. What is the wrong word here? It does not contain weakened antibodies. It contains weakened antigens. So therefore, the answer is B. Okay, moving on. 23. What are the functions of the diaphragm? What are the functions of the diaphragm and the cilia in the human gas exchange system? Okay, let's do this very quickly. We have the lungs here, and then we have like all the branches, and then you have the alveoli at the very end, right? So that is just a very simplified version. Now, and then you have the one lung, the other lung, and then you have the diaphragm. So let's just color code everything here. So the diaphragm is this big piece of muscle at the bottom. And then where is the cilia, guys? Where's the cilia? The cilia is like lining the trachea and the bronchus. Okay, so it's all here, and it's moving. It moves. It's like a little sweeping motion. It moves. So let's see which statement is correct. It says the diaphragm contracts to cause breathing in. Yes, that is correct. Remember, when the diaphragm contracts, what does it do? When it contracts, it moves down, contract. And that's important for breathing in, guys, because when you breathe in, you want to have a large volume for your lungs to expand, right? So therefore, when it contracts, it goes like that to a flat. It moves down, contracts, and moves down. And therefore, you have much more volume for the lung to expand, and therefore breathing in. So far, this is correct. Let me just do some color coding here. So this is definitely correct. Contracts is correct. And then breathing in, yes, it contracts when you breathe in, and it relaxes when you breathe out. Okay? And what does the cilia do? The cilia carries mucus to the throat. Yes, it does, because when there's some mucus here, like a clump of mucus, like a sticky stuff, what happens is you don't want this to go into your lungs because it's going to clog it up, and you're going to get infection, and you get pneumonia. It's nasty. So what you want to do with this mucus, this clump of mucus, is to get it out. And how do you get it out? Of course, you can cough, but when it's so low down in your bronchus, you can't just cough it out. So the cilia does a lot of sweeping. It just sweeps it out. Get it? So eventually, when it sweeps it up to your throat, then you can cough it out. So it's very, very effective, and therefore the answer is A. So carry mucus to the throat. So this is a very good revision there. Okay, 24. Vigorous exercise can cause an oxygen debt. Which process removes the oxygen debt? So you have to understand what oxygen debt means. Oxygen debt basically means when you are doing this thing called what? Anaerobic exercise, right? Anaerobic respiration, which could come from, it does come from exercise usually, because you're moving, right? And therefore using up a lot of the oxygen in your body. So anaerobic means no oxygen required. And when you have anaerobic respiration, you're producing lactic acid, which is bad. And how do you get rid of it? You need oxygen, because the oxygen will help to convert it back into something that is harmless. So the oxygen required to get rid of that lactic acid built up during the anaerobic respiration, that oxygen required is a debt. It's like you have to pay it back in order to get rid of the lactic acid. And that is why the answer is A. Aerobic respiration, which requires oxygen, will help to get rid of that lactic acid in the liver. Okay? None of the other answers make sense, so is A. And then finally, 25. In healthy people, which substance is completely reabsorbed into the blood from the kidney nephrons? Hope you guys still remember the nephrons, right? With the Bowman's capsule and the PCT and the DCT, the proximal and distal convoluted tubule. And really, when you think about the nephron, I hope you guys know two main things that it does, which is the ultrafiltration. Where is that taking place? Which exact location? At the Bowman's, right? At the Bowman's capsule, and also the glomerulus and the glomerulus. And then the other function it does is what? The reabsorption. Guys, please remember, it's reabsorption. It's not just absorption. It's reabsorption, because you're getting it back into the blood. If you say absorption, it's not really correct. It says reabsorption. And where does this take place? This takes place at the PCT, right? The proximal convoluted tubule. And then the question now is, what are you reabsorbing? Now you think about it. What do you want to get back into your body? Because your kidneys, which is where the nephrons are, is like your excretion organ. It gets rid of the waste. What is waste? Who here is definitely waste? And I definitely don't want it to be reabsorbed. Urea is the biggest waste ever, right? So I don't want that. It's definitely not being reabsorbed. Water and salt is not that important because you can always drink more water. Salt as well is really small and is not as important as glucose. So really, if you think about the importance is glucose. Now, why do we have to reabsorb glucose? Tell me. Why am I reabsorbing? Why can't I just pass it out in my urine, just pee it out? Because what is glucose used for in your body? For respiration. So please keep the glucose, and if it's getting lost, please reabsorb it, and hence the answer is A. So question 26 is a great question about the eye. It says, what would be the pupil size and lens shape of a person reading a mobile phone text message in a brightly lit room? So let's go through each of these, okay? So the pupil size. The pupil, first of all, is, if you look at someone's eyes, you'll see like this black hole in the center. That is the pupil. And you can see that the pupil is basically this hole there. See that? It's this hole, this gap, and it lets in just the right amount of light to focus that light onto, so here's the light entering your eyes, and then it hits the retina, and it sends that light energy through the optic nerve up to your brain so that you can interpret what you see. So the pupil lets the light in. So the function of the pupil and also the size of it is something called the light reflex. So it lets just the right amount of light coming in depending on how bright it is or how dark it is. Okay, then for the lens shape, so first of all, what is the lens? It's like this bubbly shape here. That's the lens. And the lens does this thing called focusing, or we call it the accommodation reflex. So basically it helps to focus images. What that means is, depending on where you are, the shape of the lens will change to help focus the image directly onto the retina, and therefore you'll see things clearly. If your lens don't focus properly, the image you see will be blurry. Okay, so that's why the pupil size and the shape of the lens matters to help you look at things in the most clear way. Okay, now a bit more information is, I want you to look at this person's eye and tell me which one, number one, number two, which one is in a bright room, in a brightly lit room. You see the hole in the middle, right? If there's a bright room, there's a lot of light, then what do you expect the size of the pupil to be? Would it be big or small? If there's a lot of light, you don't need the pupil to be very big. So the pupil is actually small. We call that constricted, right? It's a constricted pupil. So one here is actually a bright room, and two is a dark room. In a dark room, you want to open up the pupils as much as you can, in other words, to dilate it, and therefore let more light in so that you can see things clearly. So therefore, when you read this question, you start to narrow down things already, right? Because look, this is a brightly lit room. So brightly lit, what's the size of the pupil in a bright room? Would it be large or small? It will be small, right? So therefore, it's got to be C or D, and we can eliminate A and B already. Moving on to the lens shape now, because I want to know if the lenses are fat or thin, right? So here explains what's going on. Now, this is your eye, these two images of eye, and it all depends on whether or not the thing you're looking at is distant, means far away, or close. Because what do you notice different? What's different between these two diagrams? What is one thing that's different? Well, actually, to see two things that's different. Well, actually, a few things, actually. The first thing is the shape of the lens, right? Here, the shape of the lens is thin. Here is fat, okay? And then you also see other differences like the length of the eyeball and so on. But most important thing is the shape of the lens. So remember, guys, when the thing you're looking at is far away, like a tree in the far, far, far distance, the lens will be thin. And the reason for that is because when the object is far away, the light that bounces off the object will mostly be parallel lines. And therefore, when they enter the lens, they don't need to refract as much in order to be focused on the retina. It has to be focused on the retina. What does focus mean, guys? It means they all converge on the retina. If they converge too early, like they converge here, then the image will be blurry. If they converge later, like over here behind the retina, remember the retina is this blue line here, right? So over here or over here, this, if it doesn't land on the retina, it will be a blurry image. So your lens play a very, very important role. That's why the lens help to focus, get it? Makes things clear by changing the shape to just make the light rays converge perfectly on the retina. So when the object is far away, the lens is thin. When the object is close, the rays coming out is more spread out, and therefore you have to like bend it more. You have to refract it more. And to refract it more, you want the lens to be big and fat, because the fatter it is, the more, like, I think of it as like the more distance there is and more time there is for the rays to refract and therefore converge on the retina accordingly. Okay? Just remember, close object, fat. Distant object, thin. Okay, so now let's come back to the question and see what it says. It says here, the mobile phone. What's he reading? He's reading his mobile phone. Well, your mobile phone, is that going to be far away or close? It's very close, right? You're literally holding it in front of your face, right? So therefore, it's nearby. If it's nearby, then the shape of the lens will be fat, right? Near is fat, and therefore the answer is C. Is C, because I need the fat lens. Understand? Okay, moving on. 27 here, it says, what describes how the body maintains a constant internal temperature on a hot day? Give me one keyword, guys. If this was a like a long answer question, what is the phenomenon here? The phenomenon of maintaining a constant internal condition in the body starts with an H. What is this? What is your body doing? Homeostasis, right? In fact, that is one of the characteristics of living organisms. Mrs. H. Gren, the H is homeostasis, meaning living organisms are able to regulate their internal conditions. In this case, it's the internal temperature that we're keeping constant. That's important because we cannot be too cold or too hot because we'll die. Okay, now which of these statements makes sense? Let's just go through each of them. It says vasoconstriction increases blood flow to the skin surface capillaries. That makes no sense, because vasoconstriction means the blood vessels are constricting. If they're constricting, they're like shrunken smaller, and therefore they're going to be far away. They're not going to be close. It does not increase blood flow. It actually decreases blood flow to the surface. So it cannot be A. Let's look at B. Vasoconstriction decreases blood flow to the skin surface capillaries. This is a correct statement, guys. Right? Just read the statement. Vasoconstriction is when the blood vessels become really thin, and therefore less blood flow ends up on the skin surface. So it's a correct statement, but it's still wrong, because does this happen on a hot day? On a hot day, do you want your blood vessels to be close to your skin or far away from the skin? Ask yourself. Ask yourself. On a hot day, I want the blood vessels to be close or far away. Blood vessels or capillaries, right, should be close to surface of the skin. Why? Because I'm trying to lose the heat in my body because it's so hot, right? Otherwise I'll overheat. So that's why after PE lessons, when you just did a lot of exercise moving around, did you notice most people's faces are like red, like red? Why? It's because the blood vessels have dilated so that it's closer to the surface of their cheeks, and therefore they can lose some of that heat. Otherwise, you're going to overheat, because PE do a lot of exercise. You're actually very hot. Get it? So therefore, it cannot be A and B, because it's a hot day. Clear? Hot day, you do vasodilation. And then, keep reading. Vasodilation increases blood flow to the surface. That is correct, right? Because when the blood vessels are dilated, they're nice and fat. When they're fat, they're closer to the surface of your skin, and therefore conveniently loses that body heat to the surroundings and cools you down so that you're not overheating. So therefore, the answer is C. C is correct, okay? Great. Question 28. What would lead to a decrease in blood glucose concentration? Oh, I love a question like this. Ooh, okay. Let's see. Do I have enough space? Because this is very exciting for me. because I want to write down a lot of things for you guys. So, for each of these hormones here, adrenaline, amylase, well, they're not all hormones. Amylase is an enzyme, but for all of these things here, I want us to talk about where they're produced, where from, and then going to where, and then what they do, the function. Okay, I'm gonna have to regrettably rub out some of this because there's no space. Okay, so let's start. Adrenaline is already the biggest clue. So it is produced from the adrenal gland. That's why it's called adrenaline. Okay, adrenal gland. That's just little glands that sit at the top of your kidneys. Where are they going, guys? I need more space. Okay, so while you're thinking, please tell me where they're going. Where does adrenaline go? It goes to lots of places. Usually to places that help you do it. Is the fight or flight hormone, right? So it goes to place like your heart, right? Increases your heart rate. Goes to the heart to increase the heart rate. Lots of places increase heart rate. Okay. Next one is amylase. Amylase is produced from two places: your salivary glands and also the pancreas. Okay, but most importantly, the salivary glands. Where does it go? It doesn't go anywhere because your salivary glands is in your mouth. So whatever is produced in your mouth is going into the saliva, which is still in your mouth, right? So it's just produced locally, all right, into the mouth. And what does it do? What does amylase do? It's an enzyme because it ends with A's, and therefore it's breaking down what? It's breaking down starch. Okay, it's breaking down starch. And then moving on to glucagon. Right? Glucagon, where is it produced? Produced in the pancreas. Where is it going? It goes to the cells everywhere. Okay, lots of places, just the body in general. And what it does is it breaks down glycogen to glucose. Okay, glycogen to glucose. And finally, for D is insulin. Insulin is produced also from the pancreas. It's the opposite to glucagon. So whatever glucagon is doing is doing the opposite, right? So it's also going to the body and the cells everywhere. And it helps to remove glucose from your bloodstream. So it helps to decrease glucose in the blood. Okay? Helps to decrease glucose in the blood. And if you haven't noticed, that is the answer, right? So what results in a decrease in blood glucose? It is the release of insulin. So I'm going to repeat this again. Insulin is a hormone that is produced from your pancreas, and it is produced to bring down the glucose in your blood. If your blood glucose is too high, you have a disease called diabetes. That's not a good thing. So you need the insulin to bring down, to reduce, decrease the glucose in your blood. And that is what insulin does. Let me just ask you guys one more question. When do you think insulin is produced? Would it be produced before or after you eat? Think about what happens when you eat. When you eat, you produce, well, you're eating a lot of food, right? And they're broken down into glucose. So therefore, your insulin production will increase shortly after you eat because the food that you have eaten has broken down to glucose, and therefore your glucose in your blood is high, and you need that insulin to bring down the glucose in the blood. So it helps to decrease the blood glucose concentration. Okay, and then finally, question 29, which statement about antibiotics is correct? Okay, so we're going to do some eliminating here again. So antibiotics do not affect bacteria but kill viruses. Completely wrong. Completely wrong. Okay, because it's exactly the opposite, right? It actually kills bacteria but does not affect viruses. So this is wrong. Answer B, antibiotics do not affect bacteria or virus. That's wrong. C, antibiotics kill bacteria. And no, they do not. So basically it is D. Do you guys understand? Antibiotics only kills bacteria. They have no effect on viruses. So if you have like the flu, for example, which is a viral infection, if the doctor gives you antibiotics, you can say no thank you, because it doesn't actually help, because antibiotics does not kill the virus. It just only kills bacteria, and the flu is a virus. So question 30, the diagram shows a carpel of a flower just after pollination. What is the name of the part labeled X? This really just comes to your revision. You just have to know it. X is the pollen tube. Okay, it looks like a tube because it goes down. Okay, question 31. The table shows the percentage of sperm with different abnormalities in samples from a person with normal fertility, which is this guy here, and 2 people with low fertility. So P and Q have low fertility, okay? And then it asks questions, what are the causes of low fertility in person P and person Q? So I encourage you to pause the video just to kind of understand this question a little bit before you watch the rest of it, because it's important that you try to interpret the question yourself, okay? Right, so let's talk about this now. Now, let's draw the sperm first. So this is a sperm. What is the most important structure in a sperm? I've got the head, which is where the nucleus is, right? And then I've got this neck bit here, and then I've got a tail, and then I've got like a little nose, okay? So we have to label these properly now. So the tail is called the flagellum, which I need more space to write, okay? So this is the flagellum. And then the neck is it's not about the neck, guys. Is what's important is what's in the neck, which is a lot of what? It contains a lot of mitochondria. And I hope you know what mitochondria is used for. It's used for aerobic respiration, right? So there's the mito over there. There's a lot of it over there. And then you have the nucleus, which is the most important thing, is the genetic cargo that the sperm is carrying in order to fertilize the egg and therefore make that zygote. And this is what sexual reproduction is all about. So the nucleus is there with all the DNA inside. And last but not least is this little nose in the front, which is called the acrosome. And what is that? It is a structure that contains a lot of digestive enzymes, which is very important because when it meets the egg, remember the egg is like here, right? And it's got like this jelly layer on the outside, and the acrosome, the enzymes in here is going to have to digest through that jelly layer in order to get into the egg. Here's the nucleus of the egg, right? If you can't get through that jelly layer, you cannot get into the egg. You cannot do fertilization. So the acrosome is very important for that final step. So now let's try to understand what's going on here then. Percentage of sperm with no acrosome. That's definitely a bad thing, right? Because no acrosome, you can't even get through the jelly layer. So look at this. The person with normal fertility. Fertility means your ability to reproduce. Okay, so this normal person can just reproduce normally. It has only 2% of its sperm has no acrosome. Does that make sense? In a normal person, or shall I say in a person with normal fertility, only 2% of the sperm has no acrosome. Yeah, that's okay, right? Because 98% does have acrosome, so that's why he can reproduce normally. Get it? So I hope this is all making sense. So we need to explain person P and person Q. So let's look at person P. What is the problem with person P? Because all of these are problems. The first problem here listed is no acrosome. Then think about it. If there's no acrosome, you cannot get through the jelly layer. That's a big problem, right? The second problem is deformed flagellum. Oh dear, if you have a deformed flagellum, then you're not able to swim properly, and then you're not going to be able to get to the egg at all. And then the third one is few mitochondria. That's also a problem because few mitochondria, you don't have enough energy to do anything, basically. It's like if you don't eat, then you can't really go to PE lesson and all of that. Get it? So now we've talked about that. Let's look at what's the problem with P. P's major problem is what? 25% of the sperm has a real issue, right? Because 11% and 4% is the same as a normal person. So therefore, the 11 and the 4 here is not the problem. Get it? The problem for the person P over here, the problem for person P over here is this. Is the fact that he has a quarter of his sperm doesn't have an acrosome. So what would that mean for the person? If you don't have the acrosome, what problem will you face? Too many sperm cannot digest the jelly coat of the egg cell. Does that make sense? Too many sperm cannot digest the jelly coat of the egg cell. Yes, that's exactly what no acrosome is going to do to you, right? So therefore, it's got to be A or B. Get it? Because only A or B is talking about the jelly coat of the egg cell. So I can already eliminate C and D. That's so easy. Okay, and then let's try to explain Q now. What is the problem with Q, right? So Q, for person Q here, 2 and 2 is the same as a normal person. So therefore, it's not the acrosome issue. Acrosome is not the issue here. It's got 35% in Q has a deformed flagellum. Oh dear, that's a problem, because if you have a deformed flagellum, you cannot swim. So which one here explains not able to swim? Too many sperm cannot digest jelly coat of egg cell. No, because it's the same as this one, so that's definitely not. What about here? Too many sperm cannot move to the oviduct. That is correct, because if you have a deformed flagellum, that's the problem. Get it? So therefore, the answer is B. So I'm just going to color code this in a more helpful way. So the no acrosome in P, the consequence of that is no acrosome means you cannot digest the jelly coat of the egg. So that's the consequence of no acrosome. Whereas for Q, the fact that it has a lot of deformed flagellum means that it cannot even move to get to the oviduct. Remember, the fertilization takes place in the oviduct. Okay, the oviduct. Okay, great, amazing. All right, question 32. Then it says, which word is used to describe stem cells? Ooh, looks very straightforward. Stem cells are, okay, well, let's go through elimination process. So gametes are just, well, what are gametes, guys? Sometimes I realize that the questions that are wrong, you don't actually even know what they mean, okay? So gametes are the sex cells. Who are the sex cells? Egg and sperm in humans, right? So that's what gametes mean. So it's got nothing to do with stem cells. So it's not A, basically. And then haploid. What is haploid? Haploid is, the definition is you only have one copy of every chromosome. And that is the case in the sex cells. In other words, all the sperm cells and all the egg cells are haploid cells. They only have 23 chromosomes. 23 chromos, okay? And it's got nothing to do with stem cells either, so it's not B. But at least we know what haploid means now. And then homozygous, that is just a fancy word. It just means it's a word relating to genetics. And that's not the issue here, so it's not C. And then finally, stem cells are unspecialized, so the answer is D. Remember, unspecialized means they have the potential to differentiate into many different types of cells. Okay? So stem cells are unspecialized. That's just a word you have to know. So question 23 says, the graph shows the concentration of 4 hormones during the menstrual cycle. Which line represents progesterone? So there's a few main things to remember about this graph that you'll just make things really easy in the future, okay? Is that the one that is like a really sharp thing, almost like a horn there, that peak, that one is always LH. So A is LH. And that's because LH is around the middle of the cycle on day 14. And it coincides with ovulation. Remember, ovulation is when an egg is released from the ovary into the oviduct, and then it sits there to wait for the sperm to come and fertilize it. Okay? So the sharp one at the very top is always the highest. Zoom, that is always LH. Just remember that for a first thing. The second thing is that there's 2 humps, like this little hump and this little hump that looks like overlapping waves here. The first hump is always estrogen. This is always estrogen. And then the second hump is progesterone. And if you sort of redid your progesterone, if you did your revision, you know that estrogen, helps to thicken the lining of the uterus and therefore, prepares for implantation. And then progesterone helps to maintain the thickness of the lining. So of course you have to thicken it first. That's why estrogen goes up first before you maintain it, which is why progesterone goes up later. Okay, and then that leaves the last one, which is the pink one, the pink line, the D. D is the last hormone, which is FSH. So these are the 4 menstrual hormones. Okay, so question 34 is a great puzzle, but you can't just be good at puzzles, you have to be great at genetic puzzles. So it says, the pedigree diagrams show the pattern of inheritance of a genetic disorder in 4 families. Families A, B, C, and D. This genetic disorder is not sex-linked. And then it asks you which pedigree diagram proves that the genetic disorder must be caused by a dominant allele. So remember, a dominant allele is when you only need one copy to see the phenotype. Remember genotype and phenotype, right? Genotype is the letters. So let's say the condition we're talking about here is condition A. So you would get two copies, one from your father, one from your mother. So you could either get both big A's from, one big A and one big A from your parents each, or you would get a big A or a small a from each of them, or you could get a small a and a small a, right? So which one of these are actually dominant alleles? Dominant alleles means that you only need one copy, so one big A, dominant means capital, right? So those that have one big A, so big A or big A small a, they will have the phenotype, which is the genetic disorder in this case. So get your vocabs right, first of all, dominant and recessive. Okay, dominant means one copy and you'll see the disease. So this person has a disease, this person has a disease, and then this person does not have the disease. Now, the second thing to look at is the word sex-linked. If it's sex-linked, it means that sex means male or female, right? So if a genetic disorder is sex-linked, it means the gene that is diseased is on one of the sex chromosomes, X or Y, usually on the X chromosome. And therefore, it will affect the male and the females disproportionately. For example, the condition will just affect males. Now, I don't see that here. It says not sex-linked. Not sex-linked means it doesn't affect males or females unequally. It affects all males and females just randomly. Get it? And that seems to be the case here. I hope you know your basics, right? The square here means a male. The circle means a female. And then if it's colored, like if it's dark colored, then that means they have the condition, so it's affected. Yes? Now, the fact that I'm seeing both black squares and black circles tell me that none of these are sex-linked, right? Because if it's sex-linked, it will just affect the males, for example. So that is that. Now, before I proceed further, there's one option I can definitely get rid of. Which one is definitely wrong? Like, you know for sure that it's wrong. C is definitely wrong, right? Because C doesn't make any sense. Everyone is affected, okay? So C is definitely wrong. Now, let's go a little bit deeper into A, B, and D so that hopefully I can convince you that the answer is one of them. So let's start with A, okay? So the key here is to just sort of fill in the letters for everybody. Now, assuming that big A is the dominant allele, which is the disease, right? Because you're supposed to prove that the disease is caused by a dominant allele. Let's try to fill some of these in. Now, the white color means that no disease, right? So I can already fill in all of the white ones will be small a, small a, small a, small a, small a, small a, like this, right? Now think about what is the genotype, the letters for the remaining these three black ones. Now, this one is a black, which means that this son must have gotten one big A. And that one big A must have come from his dad, right? Because his mom doesn't have any big A's to offer. So this son would be big A, small a. Do you get it? Because he would have got the big A from his father and the small a from his mother. So I can sort of fill that in out already. And same goes for the daughter down here. This daughter got the big A from her father and the small a from her mother. So I hope you're agreeing with what I'm drawing here. Okay? Let's do D. Let's do D because just to try something different. Same thing here. So I know that all of the white ones are small a, small a, small a, right? Because they don't have the condition at all. That's why they're smiley face. And then the big one, same thing here. This grandfather here must have at least one big A because he passed on that big A to both his son and his daughter down here. Make sense? So he has one big A. The other one, it doesn't really matter. I don't really know because I don't have any information on his parents. Then this daughter here, so oops. Okay. This daughter here, what is her genotype? Can you tell me? She's black, right? That means she must have at least one big A. That there's a black circle. So she must have at least one big A. So one big A. And then she would have gotten that big A from her father. So the other one must come from her mother, which is a small a. So therefore, this is a big A, small a.
Yeah, so continue on with that logic. This son here would also be a big A, small a. A big A, small a. This woman that the son married, this circle, must have at least one big A. Agree? Must have at least one big A. And the other one, I don't know, because I don't have any information on this woman's parents. I don't know what her parents' genotype is. So it doesn't matter. But I do know that the two sons that they have, what is their genotype? Their genotype is definitely big A, and big A, big A. Yes, because they got the big A from their dad and also the big A from their mother. Okay, now they could also be big A, small a, big A, small a. Why is that? Because they definitely would have gone one big A from their mother, but their father has a small a to offer because this small a comes from his mother. You see that? So therefore, they could either be big A, or big A, small a. So hope everyone's agreeing with me so far. So now we're in a bit of a pickle because everything seems to make sense for A and D. And the correct answer is one of them. That's why I'm not going to waste time talking about B. Okay, I'm just going to focus on A and D here because B is actually very similar to A in terms of the diagram. So now it becomes very, very interesting. This is one of those higher level questions for the really smart people. Is since A and D both make sense right now, isn't it? Because everything we've drawn out makes sense. Then we haven't really answered the question because we have to pick one and it better be the correct one. So we have to kind of think of it in the opposite way now. Because if I prove that this condition is caused by a dominant allele, that means it must not be due to a recessive allele. And therefore, I have to prove the opposite. I've got to prove that it is not due to a recessive condition. So therefore, we're going to talk about recessive here. And that's a good thing because we're going to revise recessive now. Recessive means you need 2 copies to see the disease. So I'm still going to draw out these 3 options, big A, small a, and small a, small a. But if this condition is caused by a recessive allele, that means I need both copies in order to see the condition. And therefore, only the small a will have the condition. Whereas the big A's, even having one big A, is protective. So if I assume it is a recessive condition, because I have to prove it's not, right? So let's test this. Let's test that it's not. So let's test whether or not this fits into A. So going to A now, I'm going to imagine this is due to a recessive condition. So if it's a recessive condition, remember the person that's affected is always the dark color, the colored in, the black color, right? So therefore, if it's a recessive condition, the black ones now, so this one and this one, are the small a, because it is a sad face, right? Because it's recessive. So therefore, this is now small a, and that makes this guy here a what? Big A, or big A, small a? Tell me. The fact that it's white means it's not affected means that it could either be big A, or big A, small a, right? So it could either be big A, or big A, small a. However, the fact that they have a daughter that is a white one, which means that is unaffected, means that this daughter must have at least one big A. I repeat, as long as you're white, you have to be at least one big A, because one big A is protective. That's why you're white. That's why it's a smiley face, isn't it? And therefore, you can work back now. For this daughter, she got the big A from her mother, and then the small a from her father, right? So therefore, this daughter must be big A, small a, because she only got the big A from her own mother, which protected her from having the condition. That's why she's white. And then the small a she got from her father. Okay? Now, again, I don't know what this one is because I don't have any information on this woman's parents, so I don't know. But this is the bit that's supposed to make you realize A is wrong. It's because it works for both. Huh? Look! It works in the purple version, which is the dominant allele version, And it also works in the pink version, which is the recessive allele version. So how can it work for both? That means it has not proven, it did not prove that it must be due to dominant. Do you get it? Do you understand that? So therefore it cannot be A. Now I'm going to continue with this diagram here. So for D, have a look, yeah? I'm going to play the same game. So now again, remember we're testing the recessive one, right? So recessive means, just like this one here, the pink version, the black ones are now small a, right? So I'm going to do exactly that. I'm going to fill in the small a, because if this is a recessive condition, if you're affected, which is sad face, which is black, you're small a. So what is this guy here then? Tell me. She's a girl and she got one copy each from her dad and her mom. Her mom is small a. Her dad is small a. So she could only be what? She could only be small a, right? Because she only has small a from options from her father and mother. Her father and mother don't have any big A to offer. So therefore this little girl here must also be small a. And is that possible? No, it's not. Because if this is white, means she's not affected. If she's not affected, she cannot be small a. If she's not, I repeat this, if she's not affected, she must have at least one big A for the smiley face, right? But she does not have a single big A because she got small A's from both her father and her mother according to the pink version. And therefore, for D, recessive does not work. Only the dominant works. And therefore, D has proven that this is due to a dominant condition, a dominant gene. Guys, if this is a real exam, please don't waste too much time. This one question has cost us so much time. It'll cost you a lot of time. This is an exam skill moment. If one question is taking you forever, move on, do the others first, come back to it later. It's not worth your time if you take ages on it because it's just 1 mark at the end of the day, okay? Of course, if you're aiming for full marks, then you should spend more time on this, but make sure you don't jeopardize the remaining questions. Hey guys, I know that genetics question just now was kind of hard, so I have one other video on my channel where I go through a lot of similar exam questions on genetics, especially pedigree diagrams, dominant, recessive alleles, and so on. So if you go to my channel, you go to YouTube and you type in Stoic Students -123, Stoic Students -123, and then just go to the playlist. You see playlists there. So make sure you're on the right channel and then click playlists and then go to the topic videos playlist. See that? And then, as you know, blue color is chemistry, red is biology, and green is physics videos on my channel. So you see the red video that says everything you need to know about genetic and pedigree diagrams. That is the video you want to click on. And you have a lot more practice on similar types of pedigree diagram questions. All exam papers. Good luck, guys. Okay, so 35. What are correct statements about meiosis? Remember, meiosis is when you have diploid cells and then they will split in half to make a total of 4 haploid daughter cells, right? And this only happens in the sex cells, the sperm and the egg. So meiosis does not produce genetically identical cells, does not, and therefore we will rule out A and B. Because meiosis produces genetic variation, meaning that the 4 daughter cells are all different, genetically different to each other. And that's why if you are watching this video and if you have a sibling, if you and your sibling have the same parents, how come you guys don't look the same? Huh? Because your parents gave their genes to you and their genes to your sibling. How come you do not look anything like your sibling? It's because of the genetic variation produced in meiosis. It's because the parents, when their sperm cells did the meiosis division and therefore had 4 different haploid daughter cells, those different cells gave rise to genetic variation, meaning that their offsprings will all look different to each other. So definitely no for meiosis, not identical cells. And then here it says, is meiosis involved in repair of damaged tissues? No, it's not. Okay, it's not. It really is only for what? What is the purpose of meiosis? One word. What is it for? It's for reproduction. Sexual reproduction. Get it? So therefore it's not this, it's not yes. And therefore the answer is D. Okay, question 36. The photograph shows a species of fish called a leafy sea dragon. Oh, it really does look like a dragon. And the leafy sea dragons live in areas of the sea where seaweed is present. Seaweed is present. And which statements explain how natural selection caused the leafy sea dragon species to develop? So when you read this question, just before you even look at the rest of the question, just have a think, like, what are they trying to test you on? Think about the entire 2 years you spend learning the IGCSEs. What is the concept this is testing you on? Is it testing you on enzymes? Probably not. Is it testing you on Mrs. H. Green? Not really, right? Is it testing you on photosynthesis? I don't think so. What is this one testing you on? It's testing you on natural selection. Natural selection, which is all about, remember, natural selection is all about the buzzword is survival of the fittest. It means that if you have the right characteristics and if you can adapt accordingly to your environment, you have a better chance of survival. So have a look at this sea dragon here. What does it show you? This is actually a fish. It doesn't look like a fish. In fact, it looks a lot like seaweed. So why do you think the fish lives in an area that looks like this, lives in an area coincidentally that has tons of seaweed? It is a survival mechanism. It is a survival advantage, right? Because if you look like seaweed, you're just going to blend into the seaweed, and therefore you would not fall prey to bigger predator fish, and therefore you have a survival advantage. You're going to reproduce and pass on the genes that make you look like seaweed to make more offspring that look like seaweed looking fish. Yeah. Okay, so understand what is trying to tell you here. And then let's read the statements now. So which one is correct? Leafy sea dragons look like seaweed, were better adapted to the environment. Yes, I agree, because it gives them a survival advantage. So yes, that is correct. Number 2 says, leafy sea dragons that survived passed on the allele for their appearance to their offspring. Yes, because that is all what natural selection is about. It's about reproducing and passing on that allele, that version of the gene, right? And therefore you can see it in the next generation, So 1 and 2 is correct. Number 3, humans selected leafy sea dragons that look like seaweed. No, this is completely wrong, because this is natural selection. If humans did the selection, what is that called? If humans did the selection, that would be called artificial selection, which is not what this question is about, right? This is artificial selection. And this is in the sea, so humans are not involved in any of that. So therefore 1 and 2, so the answer is B. Okay. And then finally, 37 says, what is the main source of energy input to biological systems? Energy input, just the main source, right, comes from the sun. I hope you realize that, because the sun enables all of the plants to grow, okay, and then the plants will grow, and then that will give energy to whoever's eating it, such as, a cow. And then depending on if you're vegetarian or not, it will get eaten by humans as beef, and so on. But ultimately the energy source comes from the sun. Okay, last 3 questions, guys. Almost there. You're doing so well. Okay, so 38 says, which organisms remove nitrate ions from the soil? Now, this is a great diagram. Before you even look at the answer, just let's look at the diagram first, okay? I'm going to label everything. Surprisingly, not all of my students know what all of these are called, okay? So NH3 is ammonia. And then NH4 is ammonium. Ammonium is an ion. Okay, it's got charge. Ammonia is a gas, or it definitely doesn't have charge. It's just NH3. And then NO2 minus, this one is nitrite. And then NO3 minus is nitrate. So just make sure we know the names of these polyatomic ions. Okay? And I'm sure you know this one up here is called what? Please tell me. Please don't tell me you don't know what this is. Okay, that's nitrogen. So this is the nitrogen cycle. And then just revise the main points here, right? So for nitrogen cycle, the nitrogen in the air is going to go into the soil and turn into ammonia. And that is done by nitrogen fixing bacteria. You got to know all the bacteria names. That's what they expect you to know. So you have to know nitrogen fixing bacteria. That's one of them. So that is from nitrogen in the air as gas into ammonia in the ground. That's the job of nitrogen fixing bacteria. And then the ammonia here is going to get turned into nitrate. And this is done by the next bacteria, which is a nitrifying bacteria. So just know your bacteria names, okay? And then since it's a cycle, right, the nitrate is going to go back up into the air as nitrogen. And that is done by what? It's done by denitrifying bacteria. So just know all the bacteria names, basically. So that's really the three main groups of bacteria you have to know. Nitrogen-fixing bacteria, number one. Nitrifying bacteria, number two. And denitrifying or denitrification bacteria, number three. Okay, so once you know that in your head, please draw that on the exam. When you go into the exam, just draw this down before you forget it. Okay, just write it, just draw it down so that when you see a question like this, you can figure out right away. It says, which organism remove nitrate ions from the soil? In other words, this one here, nitrate, that's nitrate, right? Gets rid of the nitrate, removes the nitrate from the soil, gets rid of it and turns it into nitrogen gas, right? So it's this process. This process is done by which bacteria? It's done by the denitrifying bacteria. So the answer is B. Get it? Okay. And then 39 says, what are two reasons for organisms becoming endangered? Endangered means that there's less of them in the wild, right? This is really common sense. I'll be really happy to see this on exam because it's just common sense. So what makes something endangered, which is very sad? Habitat destruction, yes, that's correct, because they have nowhere to live. Monitoring, not really. Monitoring helps them, right? Because it makes sure that they're okay and their numbers are not depleting and so on. So not really monitoring. Education definitely not makes them endangered. It's the opposite. Education is a good thing. It makes us understand how to preserve, the natural environment and habitat preservation, and therefore education does not make things endangered. Pollution, yes, all the bad things basically. Captive breeding programs are actually good, so it's wrong. Captive breeding programs means you captive means you capture them and then you breed them to make sure that they can continue to reproduce offspring, and then you release those offsprings back into the wild to make sure their numbers are still not getting too low in the wild. Get it? So captive breeding does not lead to endangered organisms. So therefore 1 and 4. So the answer is A. Okay, and the last question. Woo! What is an example of use of genetic modification in agriculture? Okay, this is great. If I just tell you the answer, we'll explain it. Okay, so C is the answer. And genetic modification, what is that? Inserting genes, right? Inserting genes because you're modifying the genes. Agriculture is crop plants. So basically, you are inserting certain genes into the crop plants. Crop plants are plants that we grow in large amounts to feed as food for ourselves and so on. To improve the nutritional qualities of the plant. So the most famous example is golden rice, right? Golden rice is when they put insert the vitamin A gene into the rice, and then the rice that's grown, rice is a crop plant, is golden in color because of the vitamin A gene that's been put inside. And that's really good because it increases the vitamin A value, the nutritional value in the rice, which many people eat as a staple around the world, and therefore they're no longer deficient in vitamin A. So that is why C is the answer.
Follow the link in the description box below to download annotated past papers and more resources.